Okay, welcome back to our biochemistry lecture series. To watch this video, I suggest you to please watch my previous biochemistry lecture video because to understand this video, you need a lot of concepts which I explained in my previous video. Okay, so today's our discussion of this video is all about to understand the nucleotides. To understand the nucleotides. Okay, so let's begin. Our today's discussion is about nucleotides. So as you know, there are a lot of sugar molecules. So just suppose here there is just one sugar molecule. There is just one sugar molecule. And if with this sugar molecule a nitrogenous base attaches to this sugar, so then we call it nucleoside. We call it nucleoside. And if with the same sugar molecule there is a nitrogenous base attaches to it, and also with this sugar molecule, if there is a phosphate molecule attaches to it, so then we call it nucleotide. We call it nucleotide. Okay, so here the phosphate molecule, it could be one molecule of phosphate, or it could either be two molecules, or it could either be three molecules of the phosphates can attach to the sugar molecule. Okay, so that's not so difficult. Okay, so here just look there, it's a sugar molecule. So there are a lot of sugars, there are a lot of sugar molecules exist. So but for our reference, for our course, we have to just concern about two sugar molecules. So okay, so the first sugar molecule is ribose, and our second sugar molecule is deoxyribose. Okay, so both of these sugar molecules, they are 5 carbon ring structure. Both of they are 5 carbon ring structure. After discussing the sugar molecules, so now we will discuss about the nitrogenous base. So as you know, there are two kinds of nitrogenous bases. So the first one is purines. The first one is purines, and the second nitrogenous base is pyrimidines. So after learning the purines and pyrimidines, so we have to discuss more about the purines and pyrimidines. But in this video, first I want you to understand the ribose and the deoxyribose, about the sugar molecules, and then later on we will discuss about the nitrogenous bases and the further details of the purines and the pyrimidines. Okay, so now we will discuss about the sugar molecules. Okay, so as you know, there are two kinds of sugar molecules. The first is ribose. Ribose is a 5 carbon ring. Ribose is 5 carbon ring, and the deoxyribose is also 5 carbon ring. Okay, so now first we will look at the ribose, and then I will tell you how the ribose can be deoxyribose. Okay, so the ribose is a 5 carbon ring. Okay, so let's draw the structure of the ribose. This is the oxygen molecule, this is the carbon number 1, this is carbon number 2, this is carbon number 3, this is carbon number 4, and here is carbon number 5, here is carbon number 4, here is carbon number 3, here is carbon number 2, and here is carbon number 1, and this one is the oxygen molecule. Okay, so this is the ribose, this is ribose. Okay, so just look at this. On the carbon number 2, of the ribose, there is an oxygen molecule. There is a molecule of oxygen. There is a molecule of oxygen. So if we have to change, if we want to convert the ribose into deoxyribose, and if we want to look at the deoxyribose structure, so look at the term deoxyribose. Means there is a ribose structure, there is a ribose molecule with deoxy. Deoxy means you have to remove the oxygen molecule. Which oxygen molecule? It's this oxygen molecule which is attached on the carbon number 2. Okay, so now let's draw the structure of the deoxyribose. Here is the oxygen molecule, carbon number 1, carbon number 2, 3, 4, and here is carbon number 5. This is carbon number 4, this is carbon number 3, 2, and 1. So now look at the structure of this. It's a ribose with oxygen molecule, and here there is no oxygen molecule. So when there is no oxygen molecule, so it's deoxyribose. Okay, so as just now we just learned about, there's a sugar molecule attaches to a phosphate, and there are a nitrogenous base. So the nitrogenous base always attaches to carbon number 1. Nitrogenous base always attaches to carbon number 1, and the phosphate molecule can attach onto two places. It could be either the carbon number 5, or it could either be the carbon number 3. So same goes to the deoxyribose. The nitrogenous base can attach to carbon number 1, and the phosphate molecule can attach either on carbon number 5, or it can either attach to carbon number 3. So here we go with the discussion of about ribose and deoxyribose. So for the ribose, you have to understand one more thing, that's the RNA, and there is a deoxyribose, it's DNA. So if the deoxyribose, if there is no oxygen molecules, then we can incorporate this deoxyribose into the DNA molecule, and this ribose will go to the RNA molecule. Okay, so now we will discuss about the nitrogenous bases. And to understand nitrogenous bases, I would like to take help of my book, because there are a lot of structures related to the nitrogenous bases. So, but that's not so difficult. I will first draw the structures on the whiteboard, and then I will explain it to you how you can differentiate between the nitrogenous bases. And as I told you that there are two kinds of nitrogenous bases. The first is purines, and the second is pyrimidines. Okay, so just now we learned that this is a nitrogenous base. Nitrogenous bases. Okay, so the nitrogenous bases could either be purines, or it could either be pyrimidines. Okay, so now the purines have further classified into two parts. The first is adenine and guanine. And the same goes to the pyrimidines. The pyrimidines divided into three parts. The first one is cytosine. The second is uracil. And the third one is thymine. Okay, so now how you can differentiate between adenine and guanine, and how you can differentiate between cytosine, uracil and thymine. To understand this, I would like to draw the structures of these components, and then you can understand it in a better way. So I will take help of my book. Okay, first we will discuss about the adenine and the guanine. Let me draw the structures. Now I will draw the structures for cytosine, uracil and thymine. Okay, so after I draw this, I drew the structures for you. to explain it, the nitrogenous bases, there are two kinds of nitrogenous bases. The first is the purines and there is the pyrimidines. The purines have further classified into adenine and guanine, and the pyrimidines are further classified into cytosine, uracil and thiamine. Okay, so now first we will look at the purines and then we will look at the pyrimidines. So look at the purines, the adenine and guanine. So adenine always goes with guanine. Okay, adenine will always, so look at this, adenine will always look with the adenine and guanine. So look at the structures. So adenine and guanine, they are two ring structures. There are two ring structures. So look at this, the first and the second. The first and the second. And look at the pyrimidines. It's just one ring structure. Look at this. This is just one ring structure and one ring structure. So now look at again at the adenine and guanine. How you can differentiate by looking the structures of the adenine and guanine. If there is an amino group, if there is an amino group attaches, so it is called as adenine. And if, look at this, there is no amino group, there is no amino group in guanine. So there is a amino group in adenine and there is no amino group in guanine. So there are two differences. The first is that these are just two carbon rings, and the second is that you can differentiate between adenine and guanine by looking at the structures, that there is an amino group on adenine molecule and there is no amino group on the guanine molecule. Okay, so now look at the pyrimidines. So the cytosine, uracil and thiamine. The parent molecule which first made is uracil. So you cannot incorporate uracil directly into DNA or into DNA. So you first have to convert the uracil into thiamine, or you can either convert the uracil into cytosine. So you can convert the uracil into thiamine, or you can also convert the uracil into cytosine. Okay, so now look at the differences. So this is the parent molecule of the uracil, which is one ring structure, and if you want to convert the uracil into the thiamine, so look at this, there is methylation. What? There is a methylation. If you do the methylation, so then we call it thiamine. And if you do amination, then we call it cytosine. Okay, one more time, I would like to repeat it for you. So look at the pyrimidine structure. So the parent molecule is uracil, but uracil you cannot incorporate into the DNA molecule. So what you will do? So either you can convert the uracil into thiamine. And how you can convert the uracil into thiamine? It's just that you have to do the methylation. So if there is a methylation, so then it is called as thiamine. And if you do amination, so then it's cytosine molecule. Okay, so now how you can remember that the pyrimidines contain 3 molecules and the purines can contain the 2 molecules. So there is just a simple mnemonic for this. To understand this pyrimidines, so the mnemonic is simple one is CUT. C-U-N-T, CUT. And to remember the adenine and guanine, this is not so difficult. So there is a simple mnemonics for this, which is purines are pure as gold. Okay, so look at this, the first molecule of this one and this one. A represents to adenine and G represents to guanine. This is the mnemonic which I follow. If you have other mnemonics, so you can just go with them, okay? After learning about the purines and the pyrimidines, there is just there is a pathology related to the purines. So I would like to discuss it with you. A simple this is a disease which is related to the purines. So as you know that the purines contains adenine and guanine, okay, and these are two carbon ring structure. So what basically happens is that normally the purines is converted into adenine and guanine, and then the final product is uric acid or urea, you can say. So which our kidneys pee out in a urine. So when these are pee out in urine in the form of uric acid, so that's basically our normal one, okay? So that's one is normal structure. So this is normal. And if our kidneys fails to do the metabolism of this, so then the level of the uric acid accumulate in our body. So look at this. So if this process will not occur normally, so in our body, in our blood, you can say the urea or uric acid level will increase. So when the urea and the uric acid level will increase in our body, so they can be stored in our body in the form of crystals. So what kind of crystals they are? So the crystals are MSU, and MSU stands for monosodium urate crystals. Monosodium urate crystals, okay? So these are the crystals, monosodium urate crystals, which accumulate in our joints. And when these crystals will accumulate in our joints, then it will lead to a pain, an inflammation of the joints. So we call it arthritis. And the name of the disease is gout. The one and only, the famous one is gout. So basically this is a normal process, this is the normal metabolism of the purines which happens in our body, in which adenine and guanine, after doing the metabolism of them, our kidneys try to pee out in a urine in the form of uric acid. This is normally happens. But if someone's kidneys are not working in a proper way, so then what happened that in our body, in our blood, the level of the urea and the uric acid will accumulate. And how can our body store this? So it's in the form of the crystals. So the crystal name is MSU, is monosodium urate crystals. So in our body there are a lot of places where these crystals can accumulate, but the famous one is our joints, in our fingers, in our toes, or in our the knees. So these crystals can go in our joints and they cause the inflammation of the joints, and the inflammation of the joints is called as the arthritis, and ultimately we found this the gout. So gout basically happens if this process not occur completely. So today's discussion is just all about nucleosides and nucleotides. So as I explained it to you in the video that if there is a nitrogenous base attaches to a sugar molecule, so we call it nucleoside, and if there is a phosphate molecule attaches to a sugar molecule, and with the sugar molecule there is also nitrogenous base, so we call the whole structure is nucleotide. And after that we discussed about the sugar molecules. So there are a lot of sugar molecules. So there the two common one is about the ribose and the deoxyribose. So we also learned about the structures. So if you remove the oxygen atom from the carbon number 2 of the ribose, so we can get the deoxyribose. After that we learned about the nitrogenous bases. So there are basically two kinds of nitrogenous bases. The first is purines and the second is pyrimidines. If we go further in the purines classification, we get the adenine and guanine, and same goes for the pyrimidines. We got the cytosine, uracil, and thiamine. Okay, so while we discussing about the cytosine, uracil, and thiamine of the pyrimidines, I would like to discuss there is also one pathology related. There is just one pathology disease related to the pyrimidines. As we discussed that there is a gout. Gout is a disease which is related to purines. So there is also a disease. There are a lot of, not just one disease. There are just 2 or 3 diseases. There are 2 or 3 complications which can occur in related to the pyrimidines. Okay, just look at this. Look at this. As we discussed that uracil is a parent molecule. So you cannot incorporate it into the DNA directly. So what you do? You have to convert the urate into thiamine. And to do this, you need one enzyme. The name of the enzyme is thymidylate synthase. So there might be a spelling mistake, so just please ignore it. So to convert the uracil into thiamine, you need an enzyme called as thymidylate synthase. So to perform the functions, this enzyme needs folate. This enzyme needs folate, or you can either call it folic acid. Okay, so look at this. So if you have to convert the uracil into thiamine, you need this enzyme thymidylate synthase. And if you want that this enzyme perform the functions completely, your body needs the folate or the folic acid. And if there is a deficiency, of the folate or the folic acid, so this enzyme will not work anymore. If there is a deficiency of the folate or the folic acid, this enzyme will not work, and ultimately we will not get thiamine. And as you know, thymine is an important structure of the DNA molecule. The thymine is an important structure of the DNA molecule. In our further discussions in the next video lecture, I would like to discuss with you about the DNA molecule or the structure of the DNA molecule. Okay, so then you can understand that thymine is a part. Thymine is a basic part, is a basic structural component of the DNA molecule. So when there is a folate deficiency, so this enzyme will not work, and when this enzyme will not work, so then we cannot get the thiamine, and if there is no thiamine, then further we will not get the whole complete structure of the DNA molecule. So what are the complications which can occur if there is a deficiency of the folate molecule? So let's discuss the complications. Complications related to the folate or the folic acid deficiency. The first one is megaloblastic anemia. And the second one is in embryonic life in embryonic life, the fetus or the baby, the baby can develop some neural tube defects. So the baby can develop neural tube defects, neural tube defect. And if there is a neural tube defects, so what does I mean? The neural tube defects means that the baby can get problems in the development of the brain, in the development of either the brain, or it can either be the spinal cord. So after looking at this, so you might know the importance of the folate or the folic acid in your body, and also look at the embryology. In embryonic life, the fetus can also get the problems in the development of the brain and also in the development of the spinal cord. So normally what happens that if there is a woman who is pregnant, and you will see that the doctors, their physicians or their gynecologists, they give 10 times more the folic acid to the pregnant woman. And why the doctors they give the folic acid to the pregnant woman? There are just the main reason behind this, that if there is a deficiency of this, the newborn baby may get the problems related to the brain development, or the baby can also get the problems related to the spinal cord. So here we go over the second pathology related to the purines. So in this video, I just discussed about these things, and in the next video we will learn about the nucleic acid structures, especially about the DNA molecules. So I hope you will like it, and if you guys like it, just please give a big thumb up. And here is my here is my Instagram handle. So if you still have some questions related to our discussion, so you can